Welcome to this debriefing on liver tumors of the annual meeting of the European Association for the Study of the Liver. My name is Bruno Sangro from Spain. I have the pleasure to share this time with Professor Peter Schirmacher from Germany, and we will discuss what we feel are the hottest topics that have been presented in the meeting. But please make sure that you get access to the liver tree where all the information about the presentations and posters on liver tumors are presented and available for everyone. So it's a pleasure to be here. Professor Schirmacher, how would you say that the main hot topics on experimental liver tumor science were in this meeting?
Yeah. First of all, I have to say that I'm truly impressed by the quality of the abstracts and the breadth of the spectrum that have been brought up. And certainly most of the abstracts are dealing with the aspects of tumor immunology, but there are still a lot of interesting work on refining the genetic work, the profiling. There are excellent work on microRNAs, and certainly good functional studies on single genes that impact on liver tumorigenesis. Another thing that impresses me generally at EASL now is the internationality of it. Specifically in this field, we have a strong contribution of the Asian community on that. And still, of course, the majority is on HCC of the abstracts, but the interest in biliary tract carcinogenesis is rising. And we see a lot of good functional studies in the mouse. I think we still have to see the advent of the new high throughput models, somatic mouse models to model these diseases. The only thing I have to say is I would have loved to see more oral presentation, considering the enormous number of good abstracts on functional studies in liver carcinogenesis. But in generally, the impression is very, very positive.
And regarding specific topics, if you consider, for instance, benign tumors, were there any relevant presentations?
Yes, there was an oral presentation from Jean-Charles No from Jessica Zuckman-Rossi's lab. I think it's very important. We still have 10% of unclassified adenomas, and they have shown that at least part of it is due to the sonic hedgehog pathway, activation of sonic hedgehog pathway. It's linked to high bleeding risk, and this is a new molecular subgroup of adenoma. And we should compare it. It's also an abstract from France, from the Bordeaux group, and they have shown actually in their abstract that the whole subgroup of so far unclassified adenoma seems to show correlation with the arginine metabolism deregulation, that you could actually screen for it by ASS1 immunohistochemistry, and they also observed this high bleeding risk. So we are actually looking forward to better classify adenoma and to better address diagnostically the high bleeding risk of adenoma. And I think these are the main findings in the benign tumors. But there's more work on the genetic work, and I also want to point to another work from Jessica's laboratory that has gone through the profiling of HCC and shown the good correlation with the morphological patterns. So we are heading towards morphomolecular correlation in HCC and better subtyping of HCC for clinical trials and clinical management.
How about immunosurveillance and generally speaking, basic immunotherapy or basic immunology?
This is certainly the stronghold of the presentations in there. There is an excellent oral presentation from Frank Tucker's laboratory on the NK T cells. They have shown that cytokine-based activation via the CXCR6 receptor activates the macrophages, and that is involved in clearance of the senescent hepatocytes and thus is important for controlling tumorigenesis. There are further work. There's a study from Cagliari in Italy that has also shown that loss of activation of the NK cells is a negative predictor for immunosurveillance, and similar findings from the Parma laboratory have also shown the negative role of NK cell inactivation for tumor surveillance. So this is certainly a very strong aspect, and there are many other works on the immuno-oncology studies that have addressed exhaustion of the immune response and the relevance for it. The role of eosinophils, there was a study from Paris, Holland and co-workers have shown that this impacts on. Then there is the work on the polarity of the macrophages. There's work done by Eitan Galoun's laboratory. There's work done by the Geneva laboratory, Lacotte and co-workers. And there is also work from the Chopin laboratory in Mainz that have shown that polarity of macrophages, activation of macrophages, is linked to a better tumor surveillance. And this is studied in the respective mouse models. And last but not least, also the role of stellate cells in the immunosurveillance is studied. Massimo Pinzani from London, the group, and also the Leschel group from Ulm have attributed to that and have shown that inflammation-driven carcinogenesis is to a certain extent controlled by that.
Would you consider this as the interface between liver regeneration and talking about IL-6 and data, liver regeneration and carcinogenesis, or rather, how would you like to be presented?
Yeah. Especially the IL-6 signaling pathway is exactly a link between the inflammatory carcinogenesis and what we could call functional gene studies. And there is a lot of seminal work. Stephanie Ressler has shown with her group that genetic changes we frequently see in HCCs, loss of chromosome 8p, inactivates corresponding tumor suppressors. And one of the tumor suppressors, SH2D4A, is linked to the IL-6 signaling pathway. So it is involved into the regulation of endosomal trafficking of the STAT3 molecule. So this is a direct link between the genetic mechanisms and inflammatory mechanisms in the carcinogenesis. There's interesting work from NGM Biopharmaceuticals. They have shown that IL-6, STAT3 signaling activation correlates or leads to the activation of FGF19 activation. We all know that some of the targeted approaches at the moment aim at the FGF19, FGFR4 axis. So there might be an interesting correlation also with specific target genes. And also the Galoun lab from Jerusalem has shown that IL-6 activation due to inflammation and also resection, so if you activate the IL-6 signaling, accelerates HCC growth. And that's clinically very important. That means that we should maybe consider, after resection of HCC, to inactivate IL-6 signaling in order to suppress the tumor-promoting effect that is exerted by the IL-6 activation. And to extend that, there are of course a lot of other functional gene studies on one important aspect is PR3 kinase ACT and TOR signaling, that this is oncogenic, that pharmacogenomic or pharmacological inactivation of the ACT and TOR signaling pathway might be positive for HCC, and there are a couple of substances out now, they are discussed here at the meeting, that might be interesting for future trials.
As clinicians, we are profoundly in need for the search of oncogenic drivers that could be targetable. Have there been any news regarding the seek of druggable targets?
There is definitely, as I mentioned, the FGF19, FGFR4 receptor axis, which is already in the scope, phase 1 studies. There is certainly some data on the hypoxia pathway, HIF1 alpha, VEGF receptor activation. that this is certainly an interesting target for inactivation. Beyond that, no new targets beyond these which we are currently following up.
How about the non-coding genes?
That's a very interesting aspect. MicroRNAs are very important because they're modulators of the gene response in carcinogenesis, also chronic inflammation and so on. So on one end, they have the potential to be targets for pharmacological intervention. On the other hand, they have potential for diagnostics. And we see a lot of studies that point towards a potential role of specific microRNAs. MicroRNA 220 has been studied by the Geneva group to have tumor suppressive activity. Then I would like to mention that it's a group we are also cooperating with. It's the group of Ahmed Abdelaziz in Cairo, who has done quite good work on microRNA 34A, which might actually act via the NK cells. We have already talked about that. MicroRNA 484 might promote tumorigenesis. Data from the National Center for Liver Cancer in Shanghai. MicroRNA 125A, 5P, may have tumor suppressive effects. And last but not least, I think it's an interesting study, not only because it comes from our institute, but addresses gallbladder cancer, which is so far pretty neglected. And Stephanie Ressler has done a comprehensive microRNA screening, and she has shown that there is a strong tumor suppressive microRNA, that is 145, and tumor promoting microRNA, which is 575, and specifically, and all comrades, microRNA 145 acts through activation of STAT1 and represses HER2, which is also an important factor in gallbladder carcinogenesis, HER2 activation. So there are interesting links, and as I mentioned, biliary tract cancer attracts more and more attention.
Yeah, as you said in the beginning, there is an increase in interest in biliary tract cancers, and particularly in cholangiocarcinoma. Have there been any relevant news?
Yeah. Beside the gallbladder work, there is interesting work. It's done by a Korean group from Daeju, An and co-worker. They have performed RNA-seq on intrahepatic cholangiocarcinoma, and they have found two subclasses, and one of the subclasses, the inflammatory group, they found related to poorer prognosis, which is interesting because it contradicts the existing data from the Anderson-Snorri-Thorgerson work and also from Lovett's work, where they have shown also that there is an inflammatory subgroup among intrahepatic cholangiocarcinoma, but in their classification it's linked to a better prognosis. So it would be really interesting to sort out the algorithms. It would be really interesting to sort out what are the genes that contribute to that and how these different findings relate to each other. I would also like to mention Ruben Plant's work from Tübingen. He has worked on tetraspanin, and this is a protumorigenic factor in CC development as it activates proliferation, migration, and invasion. And one interesting thing we're also facing in HCC is the fact what are resistance factors, and if we are successful with therapy, why people develop resistance. That's also a question which will come up in cholangiocarcinoma. And Vaquero and co-workers from Paris have shown that a signaling pathway we know already from HCC, which is the insulin IGF-1 receptor pathway, is involved in chemoresistance in cholangiocarcinoma. That would be interesting to see, and this is, of course, also has some option for druggability. Well, Bruno, basic research is one thing. I think the real critical point is how do we get it on the road? How can we translate it to clinical application? Therefore, first of all, the question, how good are we in translation? What do you see as the challenging and the main topics this year in terms of clinical application, clinical liver cancer research?
Yeah, from the clinical point of view, as many other years, we've seen a plethora of abstracts, very good presentations, either at posters or oral sessions, regarding all the spectrum from epidemiology, diagnosis, staging, and treatment options, and refining on treatment options. But there are, I would like pretend probably 4 or 5 hot topics that were basically the potential of direct antiviral agents against hepatitis C virus inducing or facilitating the occurrence or recurrence of hepatocellular carcinoma. Second, there is a renewed interest in some serum biomarkers for diagnosis and prognosis. Third, we are starting to be able to check whether the genetic signatures and other molecular tools that were initially designed could be translated for a more personalized medicine. And finally, there have been at least 2 large clinical trials that have shed some light, and that will quite probably translate into changes in clinical practice.
I mean, DAA is one of the hot topics, and certainly among the DAAs is the question of increased or not increased cancer risk, and what do we have, how do we have to deal with that? So what are, from your side, the most important findings on that?
The first thing is the debate hasn't ended.
Okay.
And the debate will still be there for the next months and years. For instance, we have very interesting meta-analysis and meta-regression analysis from an Australian group trying to identify whether, in comparison with interferon-treated patients, hepatitis C virus-related cirrhosis or chronic infection treated with DAA resulted in a higher or lower or similar rate of HCC occurrence or recurrence. They analyzed the data from the literature, so there were no new data there. And of course, in the long run, what these meta-analyses showed is, in the long run, there's no difference. But because the question seems to be in the risk of early recurrence, there was even a signal there, because the studies with shorter follow-up did indeed showed an increased risk of HCC occurrence or recurrence. On top of that, there was an interesting presentation by a group from Israel. They analyzed more than 200 patients that had sustained virological response after DAA, and in the 18 months following the SVR, they showed that the incidence of cancer was 5%, which is high. And that includes 2% de novo HCC, 1% recurrent HCC, and 2% other tumor types. Okay. And added on that, the Spanish group that first presented on these or flagged this potential risk has now a more mature cohort in which they analyzed what was the aggressiveness of these potentially DAA-related HCCs. And among 77 patients that had complete response on HCC and had no non-specific tumors, they were able to show that there were 21 recurrences, and out of those, only 8 patients could be submitted to radical therapies, giving a signal of a potential increased aggressiveness of these tumors. So globally, the picture seems to be, if there's something related to DAA treatment, is that some tumors may become somewhat more aggressive, and then this will result in increased incidence for a short period of time, and worse patient outcomes. But this is a matter of debate. We will need to wait for a new additional data. I believe with more mature data and probably further meta-analysis, the question should be solved.
You mentioned serum markers. I mean, AFP has been around for decades. And do you see reasons to review that? Do you see new solutions on the horizon on that?
Yeah, that might be the case. One of the problems with alpha-fetoprotein as a biomarker for diagnosis of HCC was the fact that cirrhotic patients, those with active hepatitis, had usually mildly increased levels of these biomarkers, and so the sensitivity was low. And in fact, the EASL guidelines do not recommend doing surveillance with alpha-fetoprotein. But now, with the widespread use of DAA, these cirrhotic patients now no longer have active hepatitis. And this has led to the identification of ASP as a potential useful biomarker in that subgroup of patients. And for instance, a Spanish group analyzed what were the levels of alpha-fetoprotein in nearly 200 patients that achieved sustained virological response after DAA. And when they compared the baseline situation with the situation at the time of sustained viral response, the number of patients with abnormal levels dropped from 37% to only 7%. Mm-hmm. And on top of that, a group from Milan, from Massimo Colombo's group, they analyzed a short series of 22 patients that have sustained virological response and developed HCC. And they went back and analyzed what had happened to the AFP. So, and the answer was that not at baseline, but at sustained virological response, the sensitivity of alpha-fetoprotein was almost 100%. So I foresee that we will be revisiting the use of alpha-fetoprotein as a serum biomarker for surveillance.
We're all talking personalized medicine, and we have seen a lot of profiling studies and the basic. And do you see any progress in getting that on the road in real treatment now?
Lots of expectations were put on genomic analysis. Indeed, one of the main problems with that is that you have to validate in prospective series, and this is a very difficult thing to do. Interestingly, the authors of the STORM trial, that was the largest trial ever produced in the adjuvant setting of HCC, so patients that had gone through resection or percutaneous ablation were randomized to receive a sorafenib or placebo for a long period of time. Those investigators have now been able to analyze nearly 200 tumor samples that were collected. So this provided them a good platform, probably the best platform ever achieved, to understand whether the prior signatures or new signatures could be used for the prognosis of recurrence or treatment benefit. And what they found was, and there's a good part, there's a bad part. The good thing is they were able to show that phosphor-ERK immunostaining was able to identify a subgroup of patients with a different prognosis. So it shows that a very easy to use immunostaining tool that is could be used in almost any hospital could be a potential issue. Of course, this now has to be validated in prospective series, but I think it's a good achievement. The bad side is that they failed to show that prior signatures identified as prognostic for recurrence were indeed positive and useful in these prospective series. So there is a pro and a con in these validation sets.
To add a point from my side as pathologist, that's a very good argument to have tissue, to have biopsies. And like in other tumor diseases, an overall negative trial might be rescued, or at least a positive hypothesis might be generated by subgroup analysis, actually.
Absolutely.
So any further things in clinical trials you would like to mention?
Yes, I think probably the two biggest achievements in this meeting have been the presentations of two seminal works in HCC therapy. Valérie Bilgrain, on behalf of the French collaborative group, has presented today the largest clinical trial ever run on the populations of patients treated with local regional therapies. The SARA trial was a multi-center French trial that had compared patients treated with a local regional therapy that we call radioembolization or selective internal radiation therapy, SIRT, and sorafenib as a first-line treatment of advanced HCC patients. The trial had the main endpoint of overall survival and then a number of secondary endpoints. And again, there was a positive and a negative signal. The negative signal is that the trial did not prove to be SIRT to be better than sorafenib. So globally, this is a negative trial. The good thing is that it showed in a prospective way that SIRT could be used safely in this population. So up to now, we had mostly the experience for highly trained centers, and these the demonstration that in a large number of sites across France, the same good safety profile could be achieved is I think is important. Secondly, there were statistically significant differences in two important things, which are adverse events, much less frequent with SIRT than with sorafenib, and quite importantly for patients and their families, quality of life. So quality of life was sustained in the SIRT arm, and it was slightly deteriorated, as we know, in the sorafenib arm. So we now have a local regional therapy that is not better than sorafenib, but it could provide similar outcomes with good tolerability. And now we need to wait for cost-effectiveness analysis to see if indeed SIRT has any practical use in the treatment of HCC patients.
Do you see any specific subgroups that might differentially benefit from either sorafenib or SIRT?
What we have been told is that there were no difference in subgroups and analysis, but those were not presented in detail, and we will need to wait for the publication to learn about that, which is a very important question.
Yeah.
And the other final trial is the Checkmate 040. This is a clinical trial that has explored for the first time whether the blockade of the PD-1 checkpoint inhibitor could be safe and effective in the population of also of advanced patients. Prior presentations of this study had focused on the general population, dose escalation findings, etc. But here we have presented the outcomes in this specifically in the population of second-line treatment, which were the majority. So in a series of more than 150 patients, the study showed that indeed objective remissions could be observed in 19% of the patients, and a significant amount of patients had also stable disease that was very durable in time. Indeed, duration of response, the median duration of response was 17 months. And probably these stable responses or minor responses over time led to a very significant 16.7-month median overall survival. And to put this into perspective, repeatedly, very consistently, the trials run in the past in the second-line setting, similar population of patients, had shown that placebo-treated patients or best supportive care treated patients had a survival of 7.5 to 8.5. So I think this brings, again, very likely a new tool to our armamentarium for advanced case patients.
This observation is not so different from what we have seen in other tumor diseases. One observation we have in the lung cancer, for example, is that there's a fraction of about 20 to 30% that actually do worse with immunotherapy, and leading to the fact that these patients might not be amenable to even a second-line treatment thereafter because they deteriorate rapidly. Do you think the story in HCC is pretty much the same, or is it a little bit different?
I don't see the large amount of patients from not only the personal experience but also the analysis of the data in the 040 trial. There is a fraction of around 10% of the patients that really progress rapidly, whether these are very aggressive tumors that have gone through all the TACE and sorafenib, other treatments, and at that time are very rapidly growing, or the increased growth is really promoted by immunotherapy, we will not. I think we will need to wait until the results from the first-line comparative study, sorafenib versus nivolumab, in which we will have a control arm, will help us answer that very important question.
So thank you, Bruno. And again, I want to encourage everybody to visit the Liver Tree to get more information on experimental and clinical liver tumors.